I'll have to start the video with an apology. The heading is not Centriosome, it's Microtubules, Centrioles, and Centrosomes. So, starting with microtubules. Microtubules are microscopic hollow tubes, and they're made up of proteins which are known as tubulins. Now, these microtubules, they are dynamic. They undergo assembly and disassembly within the cell. So what is the function of these microtubules? First, they determine the cell shape because they form the cytoskeleton. 2. They have functions in cell locomotion and intracellular transport of organelles. And they also help separate chromosomes during meiosis. Coming to centriole. What is a centriole? One centriole is made up of 9 microtubules, like this. And this is a protein-based structure, obviously, because microtubules are also made up of proteins. A centrosome is 2 centrioles which are oriented at right angles to each other. We saw the structure of a centriole, the same thing with another centriole perpendicular to it. This is a centrosome. Now the centrosome, it lies in the cytoplasm close to the nucleus. It's not that close in this image, but they usually lie close to the nucleus. And the centrosomes get duplicated in the S phase. So what happens to the centrosomes during mitosis and before mitosis? So first, we have a centrosome there. Now during S phase, the centrosome gets duplicated. Now just before mitosis, these centrosomes start moving away from each other and they eventually stop when they reach the opposite poles of the cell. So as I said, the centrosomes move apart until they're on the opposite sides of the nucleus. Coming to the functions of the centrosomes. They form a microtubular network. So that forms the cytoskeleton of the cell. And they signal that it's okay for cytokinesis to occur. And also, once the daughter cells are formed, the centrosomes signal that it's okay for the next round of cell cycle to start. So we saw that centrosomes are duplicated in the S phase. Just before mitosis occurs, they move away from each other and they reach the opposite poles of the cell. So after this, mitosis starts. Now when mitosis happens, what happens to the centrosomes? So microtubules start growing out from the centrosomes towards something called the metaphase plate. The metaphase plate is an equidistant line from the poles of the cell. So we have one centrosome at that pole, another centrosome at this end, and between them an equidistant imaginary line is the metaphase plate. And the microtubules are growing towards that. Now we have clusters of microtubules growing away from them. Those are called spindle fibers. Spindle fibers are also made up of proteins because they are microtubules and they're present in both mitosis and meiosis. In mitosis, they're called mitotic spindles. Meiosis, they're called meiotic spindles. And these fibers or these microtubules, they attach on the kinetochores. Kinetochores are protein complexes on the centromere of the chromosome. So I'll show you an image. So supposing mitosis has started, the microtubules are growing and they are attached. Now these ones drawn in the sketch pens, they are microtubules attaching to the chromosomes. So if you look closely, we have a centromere. The centromere joins the 2 sister chromatids. And there are 2 points where the spindle attaches. That is called the kinetochore. The kinetochore lies on the centromere and that is where the microtubules and the spindles attach. And there's a last thing that I need to mention about centrosomes. What happens if centrosomes are destroyed? So first thing, their functions are signaling okay for cytokinesis and signaling okay for daughter cells to enter next cycle. So first thing, cytokinesis won't happen. So even if the cell has completed mitosis, it will not undergo cytokinesis. The second thing, daughter cells will not enter the new synthesis phase or the new cycle. So even if daughter cells have been formed, they won't move on to the next cell cycle. That is all about microtubules, centrioles, and centrosomes. Thank you for watching this video.